Thanks a lot for the nice introduction. Today is a great pleasure to be here, and I will present, together with John, that is a PhD student, and Huidong, two methods, and I call computational adventures because that's what we feel like. When you approach a method, when you approach a method development, there are a lot of up and down, and we will try to show you not a super polished story, but trying to tell you the process that we follow and also the details of the method that we end up developing. So the first story is about CRISPR tiling screen. So I just want to introduce the basic ideas. I'm sure many of you have heard about the CRISPR genome editing. So it's a system where you have a protein called Cas9, and then you have a guide RNA. And essentially, you can target this system almost anywhere in the genome. And essentially, you need to design a sequence of 20 nucleotides, and then there is a constraint. So you need to have this particular sequence is called PAM, and depends on the system you use. And for the classic system, is NGG. So you need to have this 20 nucleotide, and then followed by NGG. So with CRISPR, what you do is you introduce double-strand breaks in the genome, and then there are different repair pathways. And in the first pathway, it's called non-homologous enjoining. After the repair, you will observe deletion or insertion. And the other repair pathway is called homologous direct repair. Instead, what happens is if you introduce a donor template, the cell will try to repair this double-strand break using the donor template, and you will end up with a precise insertion or modification. So this is really nice, for example, if you want to introduce substitutions. So on the other hand, people have modified this system. So you cannot just edit the genome, but you can edit the epigenome. So the key idea is that you can create a disabled or dead version of Cas9, so you cannot cut, and then you fuse, for example, particular regulators. And you can, for example, create a chromatin repressor fusing, for example, that Cas9 with CRAB. And what happens, you will actually see after the targeting that you have an accumulation of particular histone modification that essentially will repress the chromatin. Or you can create an activator. For example, you can fuse the Cas9 with VP64 or P300 or LSD1, and you will see actually that you activate the chromatin, and you can actually, with this system, you can create enhancer element. So are you familiar with the concept of enhancer elements? No. So, we will talk about non-coding, gene, regulation. So in the genome, you have the gene, and there is a particular regulatory region it's called promoter, where different proteins can bind. But there are also other regions that are distal that are also important to regulate gene expression. Those regions, one of them is called enhancer. So essentially, the enhancer loop with the promoter, and essentially will activate gene expression. So these are, kind of switches that you have in the genome. They can activate different genes, and those are really hard to find. So the idea that, with CRISPR tiling screen, you try to explore, this genome, and you want to find those regulatory elements that are not easily annotated like genes. And it's there is this analogy. It's like, playing battleship. Imagine you have the genome, and there are some regions that are important, but you don't know where they are. But you have a way to actually perturb your system. So, with CRISPR, you can easily say, I want to perturb, this cell A1. And this is extremely easy to do. So with CRISPR tiling screen, there are 3 points. The first is what we are trying to do, with this CRISPR tiling screen. Again, we want to uncover the functional genome, and we will use tiling perturbation. And why, we need, methods is because, as John, will show you, we have, a CRISPR measurement problem. So what you measure is not perfect. So there are some distortions that, you will observe, and you need to correct for that. And how we solve this problem is using a deconvolution framework. So let's, now I want to explain you the basic steps, that you would follow to generate the data. So then, you're familiar with the data that we analyze. So the first thing that you need to do is to decide what kind of genetic or epigenetic perturbation you want to use. Again, genetic perturbation, you will introduce indels in the genome. Epigenetic perturbation, you will modify the chromatin. Then you need to select a region of interest. Where do you want to perturb, right? For example, you may want to study the regions that are important for regulating a gene, right? So what you will do, you will design all the possible guide RNA, so you can target, CRISPR here, and so on. And, you can tile up to 1 megabase. So this system is really efficient. And then, what you do operationally, when you do the experiment, you will introduce this CRISPR system, and each one will target a different location. And the idea is that only one, will go, on average, you have only one per cell, okay? And then after you do this infection, you will wait. So CRISPR, will do its job of modifying the genome. And then you can actually try to enrich for your phenotype. For the phenotype can be, for example, the expression of this gene, can be cell viability, or other phenotype that you want to study. So just like to give you like an idea of what you get exactly. So again, yes.
So tile is like, I guess, a region, not just a single point that we're modifying.
Yes.
So are we cutting out a whole region, or are we just modifying one position?
Yeah, that's a good question. So it's like independently. So if you have a population of cells, this is the region that you're trying to tile, and each cell will have one of those perturbations. So it's kind of independent experiment, right? And after you do this experiment, then you can actually do, based on your enrichment strategy, you can think to divide the population in two. Imagine the population that expresses this gene of interest and the population that will not express. And then what you do, you can actually sequence those, because when you introduce this system, like what is the sequence, so you can design primers to amplify for each cell, that region. So at the end of the day, this is like what we get. This is like the data that we want to analyze. So you have a table where for each perturbation you have a number. How many times have you observed this particular perturbation in the high population or in the low population? Okay, and then what you do is like you transform this to a one-dimensional signal, just taking the ratio, the log2 fold change, right? And then you get something that we call enrichment score. So each dot here is like one of the perturbation, and the value that you observe here is just the log2 fold change. Okay, so this is the starting point for the method development. Is this clear? Okay.
Do you maintain a notion of uncertainty? Like if you have large counts, then you know more about truth than if you have small counts.
Yeah, there are like some way to deal with this. Like there is the regularized log. So right now we don't like do this, but this is an area of improvement, right? You can say if you have low count, this ratio can explode, right? So you may want to, but right now like we don't do this in the current modeling. So just to give you an idea, like what is the general goal of this method? So you get something like this. So you have a FASTQ file. So these are sequencing reads, and what you want to obtain is like a signal that a denoise signal or deconvolve signal that tells you like what region is important, right? What region is important to regulate, for example, this gene, and some statistical test that will tell you, okay, probably this region is significant. We want to like validate. And you may want to do other operations. The problem is like from here to there are like a lot of decisions that you have to make. Like you need to align, and those sequences are short. You need some kind of scoring and normalization. There are already some problems that you highlighted. And then how do we deal with the noise of the system, and how do we deal with the statistical testing? So actually, this method has been already adopted by many groups. So it's just an overview of what people are doing with this technology. So long time ago, actually, we published a a paper where we were trying to do enhancer dissection. So you know already where is this switch in the genome, but you know that you don't need the entire sequence. So let's say you have 1 kb of sequence, and you want to find the key regulatory elements in this sequence, for example, transcription factor binding site. So you can tile this entire enhancer and trying to discover the critical subsequences of this enhancer. Other groups, and here we use a long time ago a simple hidden Markov model with 3 states. One like background, negative and positive. And then there is this paper from the Lander group where they actually use epigenetic perturbation. So as John we will show, like those epigenetic perturbations are much larger perturbations, so you can actually do an answer discovery. So you don't know where they are, and in an unbiased way, you tile 1 megabase and trying to map where are those answers. And then this other study also an answer discovery, but here you want to take a cell type where those answers are not active, and you can create those answers from scratch. And this is just amazing. So if you look the analysis, it's like everyone is doing something different. So we reason like why don't we try to think about a method that will solve all the different analysis with a unifying framework. And there are like some challenges. So the first one is that those perturbations have different efficiency. So in theory, it should be all the same, but in practice, they are not. And importantly, you remember we had this PAM sequence, the NGG, so you cannot put your guide everywhere in the genome. So this ends up with a tiling strategy where the guides are not uniformly spaced. So this is another challenge. And then like you can use genetic or epigenetic perturbations, and as John will show you, like you have different signals that you will observe. And then like what about the noise? So yeah, I just want to give you a super cartoonish illustration of this. So imagine this is your genome, and those are the regions that you want to discover. So when you like perturb your system, it's like a measurement tool that we have. So what you observe is like you kind of see there is something here, but it's like really blurred, right? So what we want to do is like we want to, as John will show, deconvolve this signal and trying to recover the original signal. And with this, I will stop here, and John can explain how we solve this problem.
Can everyone hear me? Okay. Thanks for the introduction, Luca. Really excited to come here and share some of our work on CRISPR-Time screen data. Yeah, so we call it CRISPR-Surf, or Screening Uncharacterized Region Function. And to start us off, I just want to talk about this idea of what a perturbation profile is. So we're first going to look at genetic perturbation profiles, and I'm going to turn the attention to this alleles frequency table down below. And essentially, this data right here is enumerating or showing the proportions of different genetic mutations that arise when you introduce CRISPR-Cas9, something that introduces a double-strand break and results in these variety of different indel mutations in the genome. So what's important is that each of these perturbations, they're represented in the screen thousands of times independently. So when we read out this enrichment score, what we're essentially reading out is actually a diverse set of genotypes that eventually get formulated into, at the end, our enrichment score. So how we kind of try to get at this complexity at the genotype level with just a single readout in terms of the guide enrichment score is we propose this general perturbation profile for different CRISPR technologies. So in this case, what we do is we can take the wealth of information on deep sequencing of these genetic mutations, and then we can parameterize indel distribution. So for instance, a characteristic indel is between 1 to 30 base pairs, and we can build a Gaussian window that essentially tries to describe this genotype complexity. So with, that was for CRISPR-Cas9.
What do you mean by build a Gaussian window that describes? I mean, there's a distribution of indel lengths.
Yeah.
And I guess insertion's positive, deletion's negative. You're just saying that has a Gaussian distribution, or?
Yeah, so if we take all the guide RNAs, and we measure each one of these guide RNAs, and we measure the different deletion and insertion lengths that essentially come out of these sequencing experiments, we can actually build just a window in terms of the proportion a particular base is edited away from the center where the guide is targeting. So insertions are very tricky, so you bring up a very good point. Insertions we actually model as a 1 base pair deletion, and that's because just from a practical point of view, let's say it's a transcription factor binding site that we're perturbing. To include what we think a 1 base pair insertion would do, it would actually be pulling apart this transcription factor motif. And because it doesn't necessarily affect the local spacing of base pairs outside of that insertion point, we think that it's a highly precise kind of perturbation. And it's similar characteristically to what a 1 base pair perturbation may look like when it affects the phenotype.
Are you putting in many different guide RNAs into the same system? And if so, are you able to know which guide RNA was responsible for which change?
Yeah, so yeah, that's a really important point. So in this case, this screen, all the edits from CRISPR-Cas9 guides, from all the guides in the screen, they're unobserved, okay? So we don't know what this looks like per guide. There have been many developments in the prediction of these indel distributions based on microbiology, some of it coming out of some labs at the Broad here. And I think what's really, really powerful is in future implementations, we could design a convolution from that's guide specific. So you can imagine if we're able to predict the different frequencies of edits that come from guides individually, we can encode that later on in our framework. But for now, because the edits are unobserved, we simply take the wealth of genetic editing data, and then we aggregate it to build kind of a blurred perturbation profile for all the guides from the population. And yeah, it's not perfectly Gaussian. In our manuscript, we actually show the fit, and it kind of looks a little bit sharper, like Laplace, but like a Laplace function. But I think it depends on the nuclease as well. So other nucleases like CPF1, they tend to have longer deletions, and it follows more of a Gaussian. And importantly, this framework is, you can choose, or you can even input like what you believe the convolution to be. Yeah, does that kind of answer? Okay, so in terms of CRISPR interference and activation, it's a bit of a more difficult problem to try to understand what the perturbation profile looks like. From a qualitative point of view, we know that it is much broader. And there's many, many studies that have looked at single guide perturbations from these kind of CRISPR interference and activation studies. And from this, we build this kind of general perturbation profile. Importantly, this perturbation profile is also guide dependent, and it's target dependent, depending on chromatin accessibility, different histone marks that were native at that site. But yeah, from the different data sets, we kind of have a general Gaussian window for it. So what does this mean? So if we know that genetic mutations introduced by CRISPR-Cas9 are very precise and relatively sharp, but then CRISPR interference and activation are very broad perturbation profiles, several groups have leveraged these different properties So CRISPR interference and activation are very good at enhancer mapping. And what that means is because they have such broad perturbation profiles, is that guides that are sitting even a couple hundred base pairs away from functional elements can actually feel the effects of that particular regulatory element. So CRISPR interference, even if the guide is not sitting exactly on this enhancer, it can actually feel the effects and read out that function. On the other hand, CRISPR-Cas and base editing profiles, much more narrow. So what they do, in work that has come out of Dan Bauer's group, is essentially you can actually look at enhancers that we know are functional. And then if we tile CRISPR-Cas across this functional enhancer, we can identify the critical nucleotides that essentially give rise to the enhancer's function. So this allows for better resolution of single transcription factor binding sites. And maybe you can tell about how transcription factor spacing plays a role in gene regulation. So just a brief introduction to what the convolution is. It's an operation between two functions, and essentially it describes f, which is what we conceptualize as an underlying signal, and it is convolved by some transfer function g that essentially describes some type of distortion on the signal. The convolution is a pretty broad operation that can be leveraged for many things, but in our particular case, f is our underlying function, and then g is going to be our convolver that is represented by our CRISPR perturbation profiles. So what that means is that this brings us to the idea of a CRISPR measurement problem. If we hypothesize that we have some sort of underlying genomic signal. So what this underlying genomic signal means is that if we have a score for each base pair in the genome that gives rise to a particular regulatory function, any positive effect will be an activating kind of module, and then any repressive effect will, let's say, be a silencer. And this, when we perform a tiling screen, is exactly what we want. This is the signal that we want to recover. But when we perform CRISPR tiling screen experiments, we use something that we call a CRISPR convolution. And on the top, I've outlined a perfect tool. So if any of you are familiar with the direct delta function or unit impulse, it's essentially what we describe as the perfect tool. And the perfect tool in this case is if you had a system where you could go anywhere in the genome and you could just pluck one base pair out at a time and then assess the regulatory effect of that. Unfortunately, this tool does not exist. So CRISPR-Cas9 and CRISPR interference activation technologies have some sort of variability in the perturbations they introduce into the native genome sequence, and this results in different types of distortions of this underlying signal.
Question about the uncertainty that you represent with the Gaussian width there, is that representative of your uncertainty of what you've actually targeted, or the uncertainty of the effect that it has once you targeted the metal?
The effect that it has. The effect that each sensor has. So if we think of a guide RNA as a sensor on the genome, it is simply describing that I've put a sensor here, whether or not there's a regulatory element or not. This is the proportion of bases. This is the set of bases and the proportion of edits in the population for that particular guide RNA. So in this case, if this is centered at 1, if this is a value of 1, that means that for that guide RNA in the population, that base pair is always edited. But then as you trend further from the distance of where the guide is directly targeted, the proportion of those further base pairs being edited decreases. And that's kind of what we try to capture with this Gaussian window. Okay. So just outlining some experimental realities in the data. So we've kind of shown if we have a true regulatory landscape and we hypothesize that when we use CRISPR-Cas9 and CRISPR interference activation, we can get convolved profiles. And it'd be nice if we got something that looked like this, and you could actually recover this perfectly if you knew what the G function looked like. But importantly, there are two experimental realities that make this problem a little harder. So there's the introduction of noise, but also importantly, we have targeting constraints. So as Luca briefly mentioned before, CRISPR-Cas9, or derivatives of that system, cannot be targeted to every place in the genome. It is sequence dependent and usually defined by the PAM sequence. And what's important about this kind of downsampling of signal is that it's also non-uniform. So how we think about this is how we think about how guide RNA enrichment scores arise is, again, we have a genomic regulatory signal that is intrinsic to the properties of the genome. We convolve this signal with our CRISPR convolution. We introduce, there's noise in the process, whether that's biological or technical. And then lastly, this is CRISPR tile and screen data. So this is actually real data from a screen generated from Dan Bauer's group. And then just to recap, each point is a single guide RNA and its enrichment score. And what we want to do is we want to perform the deconvolution. So we want to go from H, which is our observations in the CRISPR tile and screen data, back to this genomic regulatory signal. So importantly, in the field of signal processing, there's been several methods to be able to perform the deconvolution, inverse filtering, the Wiener deconvolution, but importantly, there's several problems in that why we can't adapt these. One being the introduction of noise, which the inverse filtering fills, but then importantly, both of these kinds of deconvolution frameworks operate in the frequency domain. So because our data points are non-uniformly spaced, and we want to preserve this information because we think it's important, and I'll kind of outline that downstream why, we don't want to interpolate our signal and then draw randomly from it. So the experimental parameters that we want to explicitly highlight in our method is we want to preserve the non-uniform spacing of data, and then we also want to allow for CRISPR perturbation profiles to change depending on the types of experiments that you perform. So how we do this is we adopt a very, very general framework called the Generalized lasso. It's essentially just least squares and then a L1 regularization on the right. And how we encode the deconvolution framework is we split up this design matrix into two matrices. One is going to encode the non-uniform spacing of guides, and the other is going to encode the convolution operation. And then for the fusion penalty, we use the first difference matrix. So that's called the fusion penalty. We believe the fusion penalty is biologically significant because base pairs, the genome has an underlying structure, has an underlying order of base pairs in a sequence. So when we try to construct this set of beta coefficients, which are the effects of each underlying, each nucleotide's function, we kind of want to keep structure. So what that means is like transcription factor motifs are usually strings of sequences that give rise to binding properties, or enhancers are strings of transcription factors that give rise to activating properties. And I will dive into kind of what each matrix looks like. But overall, we just want to perform this operation right here, where we go from noisy data to something that looks like this, which is the deconvolution.
So a lasso penalty on the difference will mean that you'll have lots of constant stretches.
Yes, yeah. Yeah, so as this lambda trends towards infinity, the standard lasso would essentially shunt every beta to 0. But then how you can kind of think about the fusion penalty, which is encoded in the first difference matrix, is it'll be a single stretch of betas at the same value at the mean of all your guide RNA guide RNA scores. And it's essentially compressing, the shrinkage is occurring around the mean, okay?
Then as you relax that penalty from that extreme, you'll get sparse jumps. So you'll have a piecewise constant function with little jumps.
Yeah, exactly. Yeah. And just to go back on that point, we've done some simulations that I won't show here, trying to play around with that fusion penalty, and we show that with certain assumptions that the fusion penalty, yeah.
Sorry, about the convolution matrix, that's known to you beforehand, right? Or are you estimating it as well?
So I can talk about ways we can try to estimate that, but we're going to make the assumption that we know from experimental data what this convolution looks like. But that's a very good point and something that we've thought about a lot. I'll talk about it when we get to that point. So just before I kind of dive into like what each part of the equation looks like, I just want to give this illustration because I think it captures just all the parts that we want to incorporate into this model. So again, if we have a true signal, we want to recover this signal. And essentially, the piece of information that we want to use are where the guide RNA is placed, what are its effects into sensing the genome, whether or not there's a regulatory element underneath it. How is that represented in the data, which are the enrichment scores? And then finally, can we use all this information and deconvolve to get a sense at what the true signal looks like? So the first part is just outlining these are y are observations, observation vector, which is determined by the length is determined by the number of guide RNAs you include in your experiment. And what we actually want to predict is a beta coefficient that is the size of the toweling space. So this is a p is greater than n problem, where which is another reason why we need to regularize this solution. But we essentially want to go from n observations to the effects of all the betas or all the nucleotides within the toweling sequence. The M, or filtering matrix, is what encodes the spatial information of where the guide RNAs lie. So I previously said we don't want to interpolate our guide RNA enrichment signal, and we want to preserve this spacing because this spacing gives us a lot of insight on the confidence in which we can call a regulatory element as significant. And the matrix just simply tells you for your given betas that you want to estimate, where are your observations located. The convolution matrix is, graphically, it's very, very simple. If you just have some sort of window or a perturbation profile that you've set, you can encode that in this kind of pattern. So going back to the question of how you could potentially estimate g of x or the convolution without making any assumptions, the way that we've tried to get at this problem is it needs to actually be done experimentally. Because when we visualize a convolution, there's two things at play. There's how wide is the convolution, the perturbation profile, and there's also how wide is the underlying functional element, okay? So a very simple way that we could actually try to estimate G is in these CRISPR toweling screens, we can actually start encoding, or we can start designing guides that target positive controls like exons. And the important thing to consider is essentially looking at how guide RNA scores ramp up on the edges of this particular regulatory element. So you can imagine just on a high level, if you have a positive control functional element and you design guides that are kind of to the side of this regulatory element, based on where it starts to increase, will give you a sense of how wide the perturbation profile is. once you're in the middle of the regulatory element, it's hard to say if it's an extremely wide element or if the convolution profile is just wide. But on the edges are well-defined boundaries that you know that you're extracting the true properties of the CRISPR perturbation rather than the functional element. Does this kind of make sense to everyone? So what's important about this is when we regress against, when this is like we're estimating the betas, each of our observations, they can be overlapping in a sense. So if you have guides that are very close together, these little yellow boxes, which tell us about what the proportion of edits are occurring in each cell, for each guide, if they overlap, they essentially are modeling shared information between our observations. And lastly, the fusion penalty. This is just a simple case, but just to show kind of what it tries to do is it tries to maintain structure. So it doesn't want like your beta coefficients to jump around when it's placed in the spacing that you believe is that is underlying the genomic sequence. So this is an unfavorable beta coefficient vector, and this is a more favorable because the number of jumps is minimized here. Yeah.
I'm not sure I understand. You have mentioned many times that you don't want to interpolate your signal.
Yeah.
But at least based on the cartoon picture that you have shown here, it seems that the location where you target your sequence, the distance between those locations is much narrower than the window of the effect. So it seems that in this regime there would be no bad consequence in interpolating the signal. Can you maybe explain?
Yeah, so it comes from, so yeah, for CRISPR interference and activation, definitely. So there are very, very little consequences of interpolation there. But with CRISPR-Cas, it's hard, I don't have a good picture to show in terms of the scale, but interpolation on a CRISPR-Cas9 screen where the perturbation profiles are only around 20 base pairs in width actually does have a lot of consequences because within a CRISPR tiling screen, let's say you use a moving average, the spacing, because it's non-uniform, even if you saturate an enhancer, there's actually many holes under the assumption that the generalized perturbation profile is true. The base pairs you would try to assess the function for, but you actually have no power to be able to find an effect there. If you did interpolate on points that are observations that are very far, I would argue that it's very different than interpolating on points that are very close if you wanted to keep that uniform spacing. But yeah, as you've said, with CRISPR interference and activation, because the perturbation profiles are super wide, when we estimate statistical power, like we essentially see that like there's no difference across. But then for CRISPR-Cas9 screens, it's very variable.
For the Cas9 case, the convolution filter is basically the convolution filter is basically discrete distribution.
Yes.
I mean, and do you see any structure in that, or do you basically, is it noisy enough? You're estimating it from read data, like at the beginning of your talk. Is that difficult enough that just fitting a parametric function is the right thing?
Yeah. So that's we've actually performed some of the analyses with just our general Gaussian window, which is also discrete. But it doesn't have some of the intricate structures of what a CRISPR-Cas9 indel distribution would look like. And interestingly, like we do get slightly different results. One important characteristic difference about the two different profiles is CRISPR-Cas9, it tends to have a sharper peak at the 0 point, most likely slightly an artifact of the +1 like Cas9 likes to have insertions of one base pair. So what we actually see from that is the perturbation profiles are even more precise than what we've parameterized. And it's not a validated element, and I probably won't show it here, but when we run it on previous data, we actually find like another GATA motif in one of Dan Bauer's old screens, and it's just something to note that we're not 100% sure that GATA motif is functional, but different perturbation profiles definitely influence the convolution and following deconvolution.
Sorry, one more question here. If you're going to get to this, then kick it down the field. But have you done this on simulated data from a model and seen how numerically stable these edges are? Because I imagine with the lasso, they could be a little unstable.
Yeah. So for the edges, yeah. Yeah. I'll get to some simulations, but I'll try to address some questions later. So one important part of this framework is selecting lambda. So it's kind of a difficult problem, but we use a heuristic, and that heuristic involves leveraging something that we do in biology all the time. And that's the fact that we perform many biological replicates when we do these CRISPR tiling screen experiments. And this kind of graph right here is a general property that if we have multiple biological replicates and then we increase our regularization, what we find is that the correlation between replicates goes up. And why that's the case is because these types of frameworks, they dominantly regularize noise at first, and then the underlying signal essentially is dominant in our final deconvolution at points where lambda is appropriate. So how we perform this procedurally is we just deconvolve replicates independently across a range of lambdas, and we've kind of determined this empirically based on like the number of guides in your system, the amount of noise that we can expect. It's just a range that we've predetermined, though it's not, as a user, if you want to use the tool, you can set that. And then we try to choose lambda based on this correlation curve. So this gets into some of the more tricky details, but we essentially perform simulations where we have multiple replicates, and we have an underlying true signal that we hypothesize. We deconvolve with our profile that we believe is related to either our CRISPR interference or CRISPR-Cas technology, and then we add noise. We downsample that signal, so the noise is added at an effect that we see in regular data, and then the downsampling is also done where we keep kind of the average base pairs per guide RNA that we see in screens, and then we essentially perform our deconvolution on the simulated data, and we try to call that element that we have in our tree signal as significant, and this is on the x-axis is as correlation increases, our ability to detect this element increases. So we just have a heuristic for determining different correlations that yield okay deconvolution profiles.
If you crank lambda way up, the correlation is going to go to 1 because you're regularizing away everything.
Yeah, so that's an interesting point.
Yeah, on your simulation.
Yeah. So one interesting thing is that sometimes that's actually not the case. And actually, so how I've tried to think about this is, let's say you have a dominant signal and in 2 replicates. And when you crank lambda super high, all of the noise gets shunted to 0. And then as your correlation, at that point when all your noise is essentially 0 and your signal's still up here, your correlation is very close to 1, right? But then what the regularization actually does is as lambda increases towards infinity, it starts pulling your signal closer to 0. So the effect of that on the correlation, actually it starts declining after some point. And then it goes, and it shoots directly to 1 when those points collapse all to 0. It's because, yeah, it's like an interesting property that we don't go that far in the lambda space. Oh, okay, yeah. Yeah, so we kind of just, yeah. So we have these optimal correlation ranges that we select for the different CRISPR-Cas9 or CRISPR interference activation modules. We assess the performance of our deconvolution with varying noise. So this is just varying the standard deviation of Gaussian error. And we show that for our deconvolution, if this is kind of like mean squared error of no signal, we recover the deconvolution. The deconvolution represents the true underlying profile pretty well. This is performance when we vary guide RNA density. Importantly, CRISPR-Cas9, which is the very, very narrow profile, is very sensitive to guide RNA density. So if you're trying to use CRISPR-Cas9 for saturating mutagenesis, just design as many guides as you can. CRISPR interference and activation, very robust. This is from average of 5 base pairs per guide to 50 base pairs per guide. It essentially shows no effect on the deconvolution. That's because it can pull signal from much further away. And lastly, this tries to get at the point of why preserving spatial information is important. We reconstruct a beta coefficient for each effect of the nucleotides. But conceptually, we know that based on where the placement of local guide RNA sit, they're going to feel different effects. So just on a high level, base pairs that are, let's say, supported by only one observation are going to have theoretically wider null distributions than betas that have more coverage or even more coverage from different independent perturbations. And how we do this is we do it in a non-parametric manner where we have some sort of set of negative control guides that can be non-targeting. We randomly sample from this set of null guides. We perform our deconvolution, and then we can construct a null distribution for each one of our betas. And then just from a practical point of view, when we run this on CRISPR-Cas9 tiling screens, they identified 5 different regions, and we're able to recover all of them. The CRISPR interference screen by the Lander group, we were able to identify all 7 validated enhancers. And the CRISPR activation screen, we identify all CRISPR-A activated regulatory elements. And then lastly, we wanted to make this accessible to the community, so we built a web-based tool in addition to a command line utility. You can see our website here at crispr-surf.pinellolab.org. And finally, just want to acknowledge especially both my advisors, Luca Pinello and Keith Jung, in addition to all the groups, including the Lander, Bauer, Orkin, and Biosca group for providing very critical experimental and statistical advice. And with that, I'll take any questions.
All right, second, computational adventure. So we will talk about single cell data, and in particular, how can we reconstruct development. So, we are talking about a method that we call of trajectory inference or trajectory reconstruction. So I want to start with the definition. What is a trajectory? So it's just a simple model that we can use to describe a possible path of transition that a cell can undergo during development or in response to stimulation. So when we talk about transitions, like we need to define what is the cell state. And there is a lot of debate about what is a cell state and what is a cell type. But for this talk, a cell state is just the current expression program of a cell or the current chromatin configuration of a cell. So anything that we can measure in a cell. So when I talk about gene expression program, you can think as a way that you can measure like all the genes that are expressed in a cell in any given moment. Okay? So there are like different analogies when you, or metaphors when you talk about trajectory reconstructions. This is from, the Waddington of T paper from, Eric Lander, and those are, pictures that, they were originally presented by Waddington. So there are like two analogies when we try to reconstruct trajectories. Imagine that, this is like your cell, and, during development, you can follow different fates. And there's one model where you have like more discrete, fates. Like you can go in this way, but as soon as you're here, you cannot jump here, right? And then there is a more relaxed or probabilistic model where you have a landscape, your cell on top of your hierarchies, like the more stem-like cell, and you want to know, given any point, what is the probability that this ball will reach any point and will end up in any particular state. So we will talk more about a method that follows this analogy. So why do we need single cell resolution if you want to develop if you want to essentially model these trajectories? The key idea is that you need to measure the single cell state, and the cell state is a property of a single cell, right? So if you have a population, what is the state of this population? It's not really clear. So if we want to model those transitions, we need essentially single cell resolution. So another important definition that we need to talk about trajectory inference is the concept of the pseudo-time. So usually when you measure your cells, like the time of the measurement is not in sync with the time of development. Let me give you an example. Imagine that you sample cells from your bone marrow, so you take blood cells, and what you will observe is that you have a mixed population. You have all the cell types. And in reality, we know that there is a underlying developmental progression, right? So you potentially start from the hematopoietic stem cell, and then you would follow different trajectory. You can, for example, go to monocytes or other lineages. So the idea is that when you measure your cells, can we find a way to unroll this differentiation axis? And the idea is that we can potentially reorder those cells, look in their similarity, how similar they are in their expression space. And if you reorder those cells, you can potentially, that's the hope, we can potentially recover the development. So this concept was proposed a long time ago by called Trapnell, where essentially they were studying myoblast differentiation, and the way to model this, to record this the pseudo-time was to do some dimensionality reduction. Then they build a minimum spanning tree where each dot here is a cell, and then they essentially follow the path from beginning, a point that you select as a beginning to an end, right? And you take the longest path. So the pseudo-time is just a numerical value that allows you to order the cell, and the trajectory is like the path that you would follow following the pseudo-time. Is this clear? Okay. So now, like as I mentioned before, the time of measurement is not in sync with the developmental time. So usually you get a snapshot measurement. Coming back to the example of the blood, you measure all the cells, for example, from the bone marrow. And what you measure are like, for each cell you have genes, and let's say you have a matrix, right? So the genes are the columns and the cell are the row, or you can, some people, they transpose this matrix. And you can measure different things. You can measure by single cell qPCR or RNA-seq, or you can also measure chromatin accessibility or methylation. So starting from this matrix, how do we reconstruct the development and all the cell states that go from a more primitive state to more differentiated state? So you want to reconstruct this tree or this graph, and also the these smooth transitions that describe this process. Okay. So I just want to give you like some really brief ideas. Like this field is really busy. Many people have proposed methods. There are like more than 50 methods available for this task, but there are like some shared ingredients, right? So if you want to like create a new trajectory method, of course you need like a matrix with your measurement. You need to normalize the data somehow, and then those two steps are really important. So if you think the original space where we operate, like you have this matrix, and each cell has like thousands of genes that you can measure. So you're in a 1,000 dimensional space. So if you try to reorder to estimate the distance in this space, like it's not really productive because you have this cursor dimensional, all the points they go to the edge. So you really need either to do variable selection. You might select genes, or you can do linear, non-linear dimensionality reduction, right? Trying to like mine this space. Then some methods they use clustering or structure fitting. The key idea is that if you want to learn this progression of states, you can use clustering to have some help in finding regions that are really stable, right? So you have a lot of cells in that state, so you can say, okay, this is a good stable state that you can use to infer things that are between two states. And then related to this, then there is some path finding. As soon as you have the structure, you can reorder the cell, and you can define what is the path. And Huidong will show you like some visualization. And we believe this is really important because if you are in more than 3 dimensions, you can really plot your space. So how can we deal with this? And I like this figure from this review paper where they say, okay, this is your gene spatial matrix. These are all blocks that we know, like you have cosine, Euclidean distance, and then a bunch of dimensionality reduction, and these are the methods that you get, right? That were published a few years ago. And then essentially the main message of this is like, well, just replace some blocks here, you get a new publication. And the steps again are dimensionality reduction, some kind of similarity or distance. Some people they use clustering, some people use graph methods like minimum spanning tree or KNN. And then for trajectory modeling, you can fit some curve in the space, or you can fit a graph, as Huidong will show. And then the cell ordering essentially is like extracting the pseudo-time. So you can propose a new method, right? Just replacing one block. So now I just briefly want to illustrate some of the key ideas that people use to evaluate methods. Because right now you can say, oh, I have a new trajectory method, like, but how well your method works. So these are I will not go into the details, but I will stick around if you're curious. So the first really popular people, they do simulations because you know the ground truth, the structure. You can compare the structure that you use to generate the data and the structure that you will infer. And like for real data is a little bit more tricky because we don't know what is the real trajectory. So the key idea is that you can use some previous knowledge. For example, for some processes, we know that there are some key genes that recapitulate the process. For example, in the original monoclonal paper, they were studying myoblast, differentiation, and they knew that ENO3, this gene, is really correlate with this differentiation path. So the game here is that you take your cell ordering, so your pseudo-time, and you correlate with the expression of the gene that you believe is important in the trajectory. And then the magic here is just like if the correlation is high, your pseudo-time is good. And there are other ideas. One that actually we propose, like sometimes you have you can use previous knowledge, but you can actually look 2 different genes, divergent genes. Imagine that you have like 2 key genes that are important to go to different fate. So there the idea is that you can actually tune this in a precision recall problem. And I don't have the time to go over this, but we have some explanation in our paper. And then other 2 things that actually are more solid. One is like time points. So if you have a process in which the time is really correlate with your differentiation, you can actually check how well your pseudo-time is recapitulating your time points. And then most recently, there is an explosion of methods where you can actually encode a barcode in the cell, and then you can actually track during development the barcode, and you can use this as a ground truth because it's something that you can actually observe. So we are taking this really seriously. This is just a small parenthesis. So we have effort with the human cell atlas. So where we are trying to, actually take this really seriously. So if you're developing or you're planning to develop a data set, a trajectory inference method, we are, currently discussing, which data set we should use, what are, the metrics that we want to evaluate, and, we have a set of state-of-the-art methods as a baseline, and, anyone can join. So if you're interested in learning more, just go to this link or, talk with me after. And the idea is that we want to make this as easy as possible. So we will, you just need to put your GitHub, page with the Docker file, and, we have standard input and output. So you just run your method, and then, you automatically get, the scoring with all the metrics. So if you're writing a paper, this is great because oftentimes they ask, oh, you need to compare with other 50 methods, right? So you can potentially use this. And with this, I will stop, and, now Huidong will show you, like, how we solve this problem and some, techniques that we use to visualize those trajectories.
Hello. Can everyone hear me? All right. Thanks, Luca, for the nice introduction, and it's a really great pleasure to be here and share with you our current project stream. It's a single-cell trajectory reconstruction and exploration and mapping. In this section, I will be mainly talking about our stream pipeline, so like how we infer trajectory and how we visualize it, and how we discover the important marker gene from the trajectories. And lastly, I will also introduce our website and available packages we have developed. So for the trajectory inference part, I will use this single-cell RNA-seq data set as example. Like in this data set, we have 1,600 cells and 4,700 important genes. And the figure here shows a very classical roadmap of a blood development system. Like in the original paper, they kind of have some visualizations. As you can see, they can plot all the genes, they can plot all the cells and highlight some cell populations and also the gene expression. But I mean, compared with the roadmap, this is not like the most straightforward or the easiest visualization, because you have to rotate all the figures. And so in stream part, we are trying to get a more straightforward trajectories, and that can illustrate the hierarchy within different systems better. So like here, this is kind of like the overview of our stream pipeline. I will go into details for some parts. Here, like starting from the single-cell gene expression matrix. First of all, we need to select the do the feature selection. Here, basically, we are selecting the most variable genes. You can see here, like based on the mean values and standard deviation, we use the local regression method Lois to trying to fit a red line here, and selected the genes that are above that fitted curve. Those genes we believe are more important in terms of the biology. And then next step, we do the dimension reduction, and afterwards we use the ElPiGraph. It's a principal graph learning method to learn the underlying structure here. This is also a very nice collaboration with Luca Pinello and Andrei Zinoviev. And after the structure learning part, we can kind of get a finalized structure. For the dimension reduction part,
Yeah, sure. Can you go back one?
Yeah, sure.
Because I just want to understand better. So your plot, why you're choosing genes above that curve as the ones you want to keep. So you're plotting mean on one axis and standard deviation on the other. So now what's the intuition for selection?
Yeah, that's a great question because like we want for the here like each dot is one gene, and for the genes that are above the because there is a correlation between the mean values and standard deviation. We want to regress that out. So we believe like the genes above that curve should like has higher variance compared to the genes like along that trend.
Imagine about skipping genes, right? So they will be the same in all the sets, right? They are not really informative for your structure. So the idea is like if you take variable genes, you will select the genes that can potentially discriminate between the different populations. So that's the key intuition. So you want to capture genes that are informative for this process.
So based on the sampling, you expect basically that the standard deviation would be proportional to the mean, and so now you want to find genes where the standard deviation exceeds what you would expect just based on the mean.
And this is not our idea. Many methods are using this. But the intuition is like if you have different population and this gene is used differently, it should be variable.
Great.
Right?
Yeah. All right. Then we can go to the next part. Like for the dimension reduction part, we are using we are using a modified version of LLE. But like here, I will just first introduce the standard LLE. It's a nonlinear method, nonlinear dimension reduction method, but it can recover the nonlinear structure using the locally linear phase. So like we have tried many different dimension reduction methods like PC or TSNE or UMAP or MDS, but we actually find like LLE works better in terms of preserving a more compact and continuous or smoother structure. This is due to the mechanism of how LLE works. Like here on the figure on the left shows the workflow of LLE. Like first of all, for each each data point xi, and first of all, it searches for the nearest neighbors, and then in the second step, it will compute the weight, which is W here, that can best linearly reconstruct xi from all of its neighbors, and by solving this constrained least squares problem. And in the third step, with all of the with the single weight vector, the low dimensional embedding vector y is computed by minimizing this cost function. Here, the t is the same as the y here. And so this is a pretty straightforward method, but you can here this classic switch row show that the LLE actually can preserve recover the nonlinear structure very well, and also it can preserve the order of all of the color.
Can I see if I understand this correctly? If I just wanted to do standard multi-dimensional, what is it? MDS, multidimensional scaling. Then I'm sort of looking at all pairs of distances and trying to preserve all of them. In this method, you're just saying for each point, it's kind of like saying I want to preserve distances between nearby points, but the way that's expressed is you express each point as a weighted average of only its close neighbors. And then after reducing, you want the weights to basically stay the same. So that even after you do this reduction, each point is still just a weighted average of its neighbors with similar weights, and you're penalizing how much those weights change.
Yeah, because like in this case, if you use the MDS, it tends to calculate all of the pairwise distance. It won't recover the nonlinear structure in the Swiss row. And but I mean, LLE works good, but here, like for the modified version of LLE, you can see the biggest difference is like in LLE, like for each point they are using it's using a single weight vector, but for the modified LLE, it's like for each point, it's using multiple optimal weight vectors. The benefit of this is can be shown like for still let's look at the spatial manifold. Like in this case, there is one rectangle missing here, and if we use the standard LLE, you can see the embedding actually has a strong distortion. But for modified LLE, it preserves the structure much better. The biggest difference is here. If you look at the cost function here, instead of one single weight vector, here it's using like multiple weight vector. Like in the original paper, they have many complex theorems to prove like how they define the SI here, like how they calculate the multiple weight vectors. If you're interested, you can check their original paper. But the difference between by using the multiple optimal weight vectors, actually it can make the dimension reduction step much more stable and robust. And so like after the dimension reduction step, like we can kind of convert a big data matrix to like you can see here the cells in the 3D dimensional space. We can already kind of see some like the very continuous structure, but how we can extract the structure here, because we want to eventually we want to use a set of the curves, which is called a principal graph, to capture the underlying structure here. In string, we are using the elastic principal graph. Given a graph, it has like the vertices V and the edges E. Here, I would like to introduce an important concept in elastic principal graph. For the K star. K star is a subgraph in G. It has K+1 nodes and the K edges. For example, like this is an example of 4 star. It has 5 nodes and 4 edges, and like 1 central node and 4 leaf nodes. And for the elastic principal graph, it's defined as a selection of the families of K stars. And meanwhile, for all of the edges, the edge elasticity moduli lambda is defined. And for all of the stars, the star elasticity moduli mu is defined. I will explain the lambda and mu in the next slide, but this is just to give you a definition of the elastic principal graph. As you can see here, like in this plot, the small dot are all of the data points, and the bigger, darker one are the graph node. Here we are trying to use the elastic principal graph to approximate the data points, and by minimizing the approximation errors. Meanwhile, we need to penalize the elastic energy, like here, which consists of the positive strings, also like the contractive strings, and also the negative strings, which is the repulsive springs. Here, as you can see, this is like actually the final objective function we are going to minimize. But before jumping to this one, I'd like to introduce another important concept here, the harmonicity, or like for any K star, when we map it, like when we do the mapping here, like the embedding of if the embedding of a central node coincides with the mean values of all of the embeddings of all of its neighbor nodes, we will say like this embedding is harmonic. So keeping this in mind, and then like for the objective function, we have like 3 important items here. The first one, it's the reconstruction error, because here the y is the node and x is the point, the data point. So here basically it's the mean squared error. And the second one is the edge. we are trying to penalize the total length. As you can see, this is like the length of the edge. And the third one, we are trying to penalize the deviation from the harmonicity. As you can see here, if the embedding is harmonic, this should be 0, and which means the central node actually is the mean value of all of its neighbor nodes. So.
What actually defines choice of such a structure? Is it assigning to every? Is it that every data point is either a center or a leaf of exactly one star, or what is it that? What assignment are we making?
No, here the data point is not the center. Here, like there are like two different concepts. Like one is a data point, and the other is a node. Like when we say the K star, it refers to the nodes, all of the nodes in that graph. Regarding the nodes here, it can be like the cluster centers of all of the points, or
Okay.
yeah.
So the centers of these stars aren't. You're choosing where those go, but then you're also assigning the leaves to data points. Is that right? Or is it just the nearest ones become the leaves, or what's happening?
Here the leaf is still a node, it's not data point.
Okay.
Yeah, so so can you rephrase that again? I'm not sure I'm getting it correctly.
So you're defining a loss function.
Yeah.
And I want to know, and so given some choice of something, there's a loss.
Yeah.
What is the choice of what? Like, I'm choosing, I have to decide I want this many stars, and this is where the center of each one goes, and this is where the leaves of each one go, and then there's some function from there that gives me a loss? Is that
Yeah.
setup?
Yeah. I think here the main idea is like because eventually we want to get a principal graph that can minimize the reconstruction error, right? But we don't want to like use too many edges, and we don't want like we want to kind harmonic embedding of the graph. So that's why like we need to add the other two items here. Imagine like we can, if we use like as many edges as we want, we will probably the approximation error will be super small, but that's not what we wanted, right? So we kind of need to use those two items kind of balance the loss function a little bit here.
Huidong, you want to explain the relation between the springs and the three parts? This will answer your the springs. In the next slide, right? Where you have the data, each data point is like, right? So explain that the springs are encoded, so they will understand.
Strings and string system.
Yeah.
Here the black springs, like the positive string, they are the contractive string. It's like the edge here. It corresponds to the edge, and the negative springs, they are the repulsive springs. It's corresponding to the stars, because as you can see, like basically, like for example, for this one, this node, it's a 3-star here. It's a 2-star here. Yeah, there are only like two neighbor nodes, and it also has a negative spring connecting its leaf nodes. So like in this case, like by minimizing this energy function, we believe like we can find the optimal embedding for this case.
So the attractive black springs want to keep the data near this graph. The repulsive ones are sort of forcing the graph to be somewhat smooth in space.
Yes.
Is that right?
Yes.
You can't be turning too fast all the time.
Exactly. Yeah. Exactly.
Right.
Yeah.
Okay.
Yeah.
Only the negative spring gives you like this harmonicity problem. So you try to find always, like configuration that are symmetric.
Yep.
So you want to like essentially describe your space in a parsimonious way. Okay, so that's essentially it.
This is all done in like a 3-dimensional or 2-dimensional embedding of the data?
Yeah, this structured learning is done in the kind of relatively low dimensional space. So it's like the 3-dimensional space or between 3 and 5. But I will introduce like we also made some extra efforts to increase the to make the structured learning work in like even higher dimensional space, like 10-dimensional or 20 or even more. I will introduce that later, and you will see the different strategies there. Having defined the elastic energy, we can already kind of evaluate evaluate different topologies here. But like in stream, we want to explore when we explore the data point space, we want to get like different topologies. Here, there is it's a graph grammar based approach. The graph grammar is basically also called a graph transformation or graph rewriting. It's a predefined set of graph transformation, and it also contains different operations. Here, like this figure shows like two simple operations. Like one is bisect edge, and the other one is add a node to another node. And you can see starting like this is a 2-star, right? This is central node, and it's a central node with 2 leaf nodes. And we can do like the operation 1 is bisect edge, the operation 2 is also bisect edge, and the operation 1 is add a node, and the other 2 are the bisecting edge. Imagine like if we keep bisecting an edge, eventually it will become a principal curve. But by like using the operation adding a node to a node, like we can eventually get a principal tree. So the process is like after the first round of operation, we can, based on the energy function, we can pick the optimal topology. And then again, in the second round, we do the different operation again, and then we can get different kind of a topology, and we will pick the optimal one based on our energy function. And this process will continue until a definite number of nodes are inserted. So then let's back to the let's go back to the stream pipeline. During the structured learning method, we need to first of all we need to seed the initial tree structure, and then we use the graph grammar to generate different topologies, and then use this elastic graph energy function to pick the optimal one. And by adding the nodes sequentially, actually, we can eventually get optimal finalized tree structure. But this because we cannot really explore all of the possible topology, because that will be a huge amount of structures. Here we are using a kind of like a gradient descent, like a greedy approach. So which means we cannot really guarantee the global optimal structure. That's why the first step, the seeding structure, is a very important step. During the seeding structure, we are using affinity propagation. It's a clustering method based on the method passing and trying to find a set of exemplars that can be best representative of all of the other data points. And then after this clustering step, we can kind of get an initial minimum spanning tree, and then we will get the node positions and also and also the connection between all of the nodes. So this information will be used as the initialization of the principal graph learning. And also we did we made some extra efforts called adjusting the final tree structure to make it more suitable to the single cell analysis. This is kind of like the structure we got after the ElPiGraph. But sometimes, as you can see here, we can already kind of capture the main structure here. We can find 2 bifurcation events here. But if we zoom in, looks like sometimes the figure on the left are are the principal graph with all of the cells, and the one on the right is the same graph. It's just I'm not showing all of the cells. Instead, I'm showing all of the nodes and the edges. So the problem here, like sometimes the branching point can, I mean, it's okay, but it's not like ideally captured, because we are hoping like the branching node should be here instead of here. So we do some extra steps like optimize the branching point by adding some nodes locally instead, because by doing this step will be super fast, and we just add more nodes by adding like more nodes locally around the branching point, so we can kind of pull the branching node to a more accurate position. And also, if you look at here, because when we calculate the pseudo-time, we need to measure the distance from the root node. And you can see here some cells like this branch doesn't really like reach the further cells. But so in that case, when if we use this graph to calculate pseudo-time, like all of the cells here will be assigned the same pseudo-time. So in that case, we cannot really distinguish those cells. So we also have some like extra grammar to extend the leaf node by adding like some extra node to trying to reach the further cells. In that case, we can calculate the pseudo-time more accurately. And also we have some extra steps like pruning the branches, like we can remove the branches without too many cells. And also an important part is like the shifting the branching node. Because when we do a structured learning, the branching, the branching node will fall in the center of the data points. But sometimes, like for example, in the single cell analysis, if the data is kind of sparse and in the center area, actually you can kind of, there may be like no cells around. And if we treat that branching node as the real branching node, that doesn't really make sense biologically. So like in that case, we can shift the branching node to a denser area or the area with more cells. So in that case, it would then like make more sense in terms of biology. And after the structured learning part, we can already get a set of curves. But this is still like in kind of a high dimensional space, like a 3 or 5 or even 4 or even 5. How should we explore and visualize it? Because this is not the easiest way to see the structure. First of all, we do like the force-directed layout, but here we preserve two important two pieces of important information here. Like one is we preserve the length of the branches. Like here, the length of the branches is the same as the length of the curve, and also we preserve the distance from the cell to the branches. So this can give us some confidence, like give us some confidence on the assignment of all of the cells. If like one cell is super far from the branch, we will probably say like, oh, maybe this assignment is not that accurate. And after this work, like we can just pick one. We need to like here, because we have some fact sorted cell labels, so we can say, oh, it may start from here. So they say like we use some. They say just we are just trying to rearrange the all of the branches using the breadth first search. So in this way, it's like a subway map. We don't care about the distances between the branches. We care more about the like the destinations. So like from the stem cell, like how they will differentiate into different like more committed cell types. But this is and it's still the same, and this is cool. But when it comes to the large data set, there will be like so many cells overlapped together, and in this case, it's very hard to tell how many cells we have here. So we need to, for large data set, we need to like add one more dimension to to kind of indicate the number of cells. Here we are the density here is like the width of the branch is proportional to the number of cells. And once we put back all of the annotations, and we can easily keep track of all of the different cell types, and we can also see how the composition of different cell types changes during the differentiation. And yeah, we spent a lot of time actually making the stream plot. I will like show you just to kind of give you an idea like how the stream plot is made. Like first of all, we need to use some like sliding windows, and we can like in each sliding window we can kind of get the number of cells, and we need to normalize them, and we also need to calculate the branching node, and then we use the linear interpolation and to get a very first draft of the figures, and later to make it smoother we use the Savitzky-Golay filter. So in this case we can get a much smoother figure. Like the one on the left is the ensemble of all of the cell types. The one on the right is just we are highlighting one cell type. And afterwards, we can kind of get a different polygon, and we just need to fill them in using the customized colors. And for the genes, it's the same, but we need to. It's like first of all, we actually need to make a picture. It's a matrix. And we need to keep the information of the cell numbers and the cell count, and also we need to calculate the average expression of one gene in each sliding window. And then, like based on this coarse image, we do some bicubic interpolation, and eventually we need to clip the stream plot out of this image, and we can get the final gene expression stream plot. So this is like just some visualization. I will skip that. And also using stream, we can discover some divergent genes which that are differentially expressed between different the sub-branches, and we can also discover the transition genes if you like walk along one branch, like how well the gene expression correlates with the one particular path. And we also can find some leaf genes that are really specific to one branch. Then I just like this is just some example like the differentially expressed genes and transition genes and all of the leaf genes here. And as I mentioned earlier, like this structure is learned from like a relatively low dimensional space. Like we have from 2 to 5 dimension. But what would happen if we increase the dimension to like 10, 20 or more? Because like with more components, with more dimension, we will have more power to discover some details. If we still use like we actually spend a lot of time like in trying to solve this issue, and if we finally realize the seeding structure actually is the key here, because like in the low dimensional space we can use this strategy, but in higher dimensional space we tend to get like too many, because the data points are way more sparse, and we tend to get too many clusters, and the minimum spanning tree will give you like too many noisy branches, which will make the later structural learning more challenging. So here, like we are using a different seeding strategy for the high dimensional space learning. Let's say like after a dimension reduction, we have like H components. And first of all, we only pick like the top L components, and then we follow the same structure as we did in the low dimensional space. And afterwards, we need to run the first round ElPiGraph, so we can kind of get a polished principal graph in the low dimensional space, and then we need to map the principal graph to the higher dimensional space. But what we can, how should we map that? Because this graph is learned in the low-dimensional space, so there will be some coordinates missing if we want to go to the higher dimension. But luckily, we have all of the cell informations, and we also have the cell edge assignment information. So we can use all of the we can try to make good use of the cell information to infer the missing values in the high dimensional space. And after this step, we can kind of get the initial node information and edge information, and then we run the second round elastic principal graph in the high dimensional space. So I will show you some results here. Like this is what we can get in the low dimensional space. But when we increase, let's say increase the dimension from 4 to 20, we don't want the branch explode and we will get a super messy. If we use the simple seeding strategy, it will the branch will explode and it will kind of look very messy. And what we want to do here, we want to pull out more detailed branches, but in the meantime, we want them to be still maintained. Let's say for the macrophage cell here, when we want to pull out more branches, we still want them to be maintained under the same parent branch. So by using the new seeding strategy, you can see when we go to the higher dimensional space, the structure is the global structure is still well maintained, but in the meanwhile, we can pull out more detailed branches. Like here for the lymphoid branch, we can get the T cell, B cell, and the natural killer cells, and also for the other cells, for the macrophage and neutrophil cells, we can also get more details. And similarly, when we do that in two-dimensional, we can only get a simple bifurcation, but if we increase to 10, we can get way more detailed branches, and those are reasonable in terms of biology. And all right, and lastly, I will just show you our website. We have an interactive website. For Stream, we want to make it really user-friendly and accessible to the general public. So actually, John did a lot of work for developing this amazing interactive website. And so it's if you are not really familiar with all of the data analysis, you can just use our website. It should be very, it's this is a simplified version of the stream analysis. And if you are an advanced user, and we will suggest you can use our bioconduct package, because in this here, you can do the step-by-step analysis and get a more strict analysis. And we also have the Docker image-based command line available. So we are really spending a lot of time and efforts in the software development part. And so we just want to make the algorithm or the pipeline really easily accessible to all of the users if you are interested. And yeah, I will just stop here, and I would like to thank everyone who helped me in this project, and especially my supervisor, Luca Pinello, for his great guidance and support, and John for this amazing developing this amazing website. And especially I would like to thank Andre and Luca for their great help in the structured learning part. And yeah, just thank all the people, and without their help, it's impossible to finish this project. Yeah, I'm happy to take questions. Thank you.